Alright guys, in this little screencast here, I'm going to show you some of the more kind of advanced DNA structure ideas. But don't get worried here because this isn't necessarily something I want you to memorize and know, but on the other hand, I think by watching this and thinking about it a little bit, it will make some of the things that we're going to do later on, like when we get into replication and transcription and translation, will make it make a little bit more sense. So bear with me here for a minute or two and just check some of this out. Okay, we have four bases here. The four bases that that you're learning about: adenine, guanine, thymine, and cytosine. So here's the trick. They fall into two different classes. One of those classes is the purines, and the other class is the pyrimidines. So we're going to look at what makes something a purine and what makes something a pyrimidine very quickly here. Now, to start, let me just start by saying this. As I was watching and thinking about what I wanted to watching some YouTube videos. See, I do that too, and thinking about what I wanted you guys to take away from this. One of the guys that was doing a presentation came up with this little memory device, this little mnemonic device, and he called it Puga 2. And I think this is great because what he's trying to do here is make a way that you can remember which ones the purines are. So a Puga 2, the Pu gives us purines because they both start with a P, but purines start with the Pu, right? And then the Ga, points out that it's adenine and guanine. All right, so adenine and guanine right there. Now it says adenosine here, but we're just going to call it adenine. So adenine and guanine. And then the 2 indicates something kind of special here, is that these both have 2 rings. And I'm just going to circle these 2 rings here so you can see that. So one of the things that I'd like you to know and put down in the notes and think about is that Puga 2, or the purines, have 2 rings and they're adenine and guanine. So then our pyrimidines have 1 ring. So if we look here, we can see they each just have 1 ring. And those are our thymine and cytosine. So as we're looking at this picture real quick, also notice here it says ribose. And what that means is that right here off of this bond, we're attached to that ribose sugar, whether it's a deoxyribose in the case of DNA, or as you're going to find out soon, in the case of RNA, it's just a ribose sugar. All right, so let's go on a little bit. So here's another picture of our 4 nucleotides together. And these are 4 nucleotides, so I have a nucleotide right here. I have a nucleotide right here. I have a nucleotide right here, and I have a nucleotide right here. And keep in mind, the reason those are 4 nucleotides is because I have my phosphate group, or what we have been calling an acid. I have my deoxyribose, or my sugar. All right? And then up here, I have my 4 bases: adenine, guanine, cytosine, and thymine. So notice when they draw the purines, the adenine and guanine, remember Puga 2? The 2 purines, Pu, are guanine and adenine, and they each have 2 rings, and they've drawn them that way. And I just put that in there so you could see that. Moving on a little bit, again, I have my purines and my pyrimidines. But we know that adenine always bonds with thymine, and cytosine always bonds with guanine. So check this out. Put it up here again. Puga 2. What are my 2 purines? Guanine and adenine. So right here are my 2 purines. And notice something. If I look at this complex chemical structure, which might not mean anything to you, I mean, we've studied carbon and we've studied hydrogen and we've studied how things bond together, and you made models earlier this year. But what's really cool here is, look, 1, 2, 1, 2. So the adenine and guanine in this picture both have 2 rings. And I'm going to change my color of my pen here real quick and just show you that over here on thymine, I have 1 ring. All right, and on cytosine, I have 1 ring. So basically what happens is a purine always bonds with a pyrimidine. So the purine adenine always bonds with the pyrimidine thymine, and the purine guanine always bonds with the pyrimidine cytosine. Another thing I wanted to point out to you in this little image here real quick is right here, and I'm going to try, if you just bear with me 1 second. I see what I did. I was trying to make my pen a little skinnier, but not so. There. If we look right here between the cytosine and guanine, and right here between the adenine and thymine, they're bonded together with hydrogen bonds. So let me ask the question. Here's a question for you. Which one is stronger? Well, count them. How many hydrogen bonds do you see between cytosine and guanine? Right, 3. How many do you see between thymine and adenine? Correct, 2. So this bond, by what we know in chemistry, should be a stronger bond than the thymine and adenine because it has 3 bonds. All right, so let's move. Okay, so the last thing I want to touch on this little screencast is the idea that the 2 DNA strands run let me get my pen on, run anti-parallel, and I always forget, is there one R or two? One, parallel to each other. All right, so if we look at this picture of a DNA, we see that there's an arrow going like this, and we see that there's an arrow going like this. And you might ask yourself, why is that? Well, how do they determine these are running anti-parallel? So parallel, they're side by side, but they're side by side, and one's upside down, really, as compared to the other one. Why does that happen? And if you look at it closely, what might pop out to you is like this oxygen, this oxygen, this oxygen, they're all pointing up, and this oxygen, this oxygen, this oxygen, they're all pointing down. And basically that's it. That they're anti-parallel, and if we look at it, we can see they're upside down. Now we have a way of determining which way they're running, and we say they run from 3' to 5'. And how do we know that, or how can we tell that? And I think it's kind of important to point it out here, but I don't want to go too in depth into it, because if I go too in depth, then it's not going to make sense. But if I give you just a little bit, hopefully it will help you when you get into replication and start looking at how replication happens. So I want to take just this sugar molecule right here and redraw it. So if I put my oxygen and I have my carbon ring coming off my oxygen, and then this comes up and there's a carbon here. Now I'm just going to draw a kind of a squiggly line to a box here. And I want you to understand that that's my phosphoric acid. And I'm going to draw another squiggly line here to a box, and that's another phosphoric acid. Now, of course, that would have a squiggly line to a carbon that's connected to an oxygen and a deoxyribose, and it would have one coming down off too. Here's the deal. When we number our carbon atoms in a deoxyribose sugar, these are all carbons where these lines meet. So there's actually 5 carbons in that sugar. This is the first carbon, second carbon, third carbon, fourth carbon, and this is the 5th carbon right there. Look what's happening. This phosphate on the bottom is connected to the 3rd carbon. This phosphate on the top is connected to the 5th carbon. They are going 3' to 5'. And then over here, since the oxygens are facing down, over here, since the oxygens are facing down, we have 3', 5'. You can't see that. 3', 5', 3', 5'. So really what's going on is 3', 5', 3', 5', 3', 5', and the same over here. 3 to 5, 3 to 5, 3 to 5. So that's why this one is going down. That's why this one is going up. That's why they are anti-parallel. Thanks.